Good afternoon. My name is Toby Astle, and it's a pleasure to be with you today, acting as the chair for the CASP Education and Training Working Group. It's a pleasure to introduce the next four speakers who will be speaking about heavy metals in cannabis. First, we have Robert Thomas of Scientific Solutions. Rob will be speaking about regulating heavy metals in cannabis, what we can learn from pharma. Following Rob, Kaveh Kehen, CEO of Avian Interchem Scientific and Chairman and CEO of Sigma Analytical Services, will be speaking about heavy metal analysis in cannabis and hemp flower and derived products, a Canadian perspective. After Kaveh, Aaron Hyman will be speaking about the validation of various metals in cannabis and cannabis-derived products by microwave digestion and ICP-MS analysis. Aaron is PerkinElmer's inorganic product line leader. And lastly, Ben Armstrong, Lab Director of Juniper Analytics, will be speaking about a perspective of a compliance testing laboratory adding heavy metals into their testing suite. Sit back and relax. Following the session speakers, we will have 30 minutes of live Q&A. So please enter any questions you have into the chat box as we go through. Thanks again.
Good afternoon, everyone. Welcome to the AOAC Heavy Metals in Cannabis Work Group training session. My name is Robert Thomas, and I run my own consulting company, which focuses on the educational needs of the cannabis industry. I'm going to kick off the session with a talk on regulations for heavy metals in cannabis and how the industry has been predominantly guided by the pharmaceutical industry. So let me dive right in by giving you an overview of my talk. I'll kick it off with the current status of heavy metal regulations of cannabis and hemp, particularly the limitations of a fractured state-based system. I'll then focus on how the pharmaceutical and dietary supplement industries approach regulating elemental impurities and how they abandoned the 100-year-old semi-quantitative colorimetric method and replaced it with modern instrumental techniques. I'll also offer my perspective on what the cannabis and hemp industry can learn from this process and give my thoughts on how the industry should move forward with federal oversight around the corner. So where is cannabis legal in the US today? Here's a map of the US with light green representing states where medicinal cannabis is legal and dark green where both recreational and medicinal is legal. It is legal for medicinal use in 36 states, with 14 of them also allowing for recreational use. However, many of the other states are going through the legalization process, so this map could look very different in 12 months. Most of the 36 states have heavy metal limits. The majority of states just specify 4 heavy metals known as the Big Four: lead, cadmium, arsenic, and mercury. Maryland adds chromium, selenium, silver, and barium, while New York adds nickel, chromium, antimony, zinc, and copper. Oregon has no heavy metal limits at all. Let's take a more detailed look at some of the individual states. This is a table of heavy metal maximum limits for 7 different states: California, Colorado, Oregon, Maryland, New York, Connecticut, and Massachusetts, compared to United States Pharmacopoeia Chapter 232 permitted daily exposure limits, often called PDEs, for elemental impurities in drug products, together with USP Chapter 2232 PDE limits for dietary supplements, and the American Herbal Pharmacopoeia limits for heavy metals in botanical and herbal products. A couple of points to emphasize: USP Chapter 232 defines a total of 24 elemental impurities based on the drug delivery method, which is oral, parenteral, or inhalation, while Chapter 2232 only defines the Big Four heavy metals in dietary supplements. So in this table, 12 of those elements have been included to reflect the Big Four plus the additional 8 elements in Maryland and New York. It's also important to emphasize the Chapter 232 limits in this table are based on microgram per day. So to know what's allowable in the actual drug or supplement, these limits must be recalculated based on the suggested daily dosage for that particular drug or supplement. For example, if the maximum dosage is 10 grams per day, these limits must be divided by 10 to convert them to microgram per gram in the drug. Let's take a closer look at 3 of the states which emphasize the fractured nature of these regulations. Let's begin with California. California was the first state to regulate cannabis in 1996, and as a result, it is considered the gold standard with regard to cannabis regulations. California limits are defined in microgram per gram in 2 different categories: inhalation cannabis products and all other cannabis products. So these are the same as USP Chapter 232 PDE limits for inhaled and oral drugs respectively, based on 10 gram of consumption per day. It's also worth pointing out that even though pharmaceutical PDEs are based on 10 gram suggested dosage per day, we really have no way of knowing in what quantities people consume cannabis products. Here are the limits for Colorado, which are a little more complex than California. Colorado limits are categorized by heavy metals of interest in the first column, acceptable limits in the second column, and products to be tested in the third column. The acceptable limits are defined in part per million in the product and not microgram per gram. Besides inhaled and oral, they also include limits for transdermal products by the skin. The limits for inhaled products are based on USP Chapter 232 PDE levels for inhalation drugs, but the limits for oral products are based on USP Chapter 2232 for dietary supplements. However, the topical values are based on FDA limits for heavy metals in cosmetics. Note these values are based on acceptable limits for 1 gram of intended use and not 10 gram as in California. Here are the limits for Connecticut. They're defined a little differently because they are based on microgram of heavy metal contaminant per kilogram of user body weight per day. So it's very difficult to get a good understanding what this means with regard to maximum daily consumption, because it's only based on body weight of the consumer. However, in toxicology studies, very often a typical body weight of 60 to 70 kilograms is used, but you still need to know what is the recommended consumption per day of cannabis product to make the calculation of the heavy metal maximum allowable limit. So let's talk more about the PDEs defined in USP Chapter 232. Here is a list, here's a full list of USP Chapter 232 PDE limits, which are based on maximum limits in microgram per day for 24 elemental impurities in the drug compound per delivery method. So for a suggested maximum daily dosage of 10 gram, these limits should be divided by 10. Note, Chapter 232 limits are the same as the ones defined in ICH Q3D guidelines for elemental impurities. For those of you who are not familiar with the ICH, it is an international council for harmonization of technical requirements for pharmaceuticals for human use, which is a consortium represent the global pharmaceutical industry, including the United States, European, and Japanese pharmacopoeias. Here are the USP Chapter 2232 PDE limits for dietary supplements only. It should be emphasized that this chapter also addresses different forms of arsenic and mercury because of the potential of different arsenic species in plant-based botanical supplements, as well as the potential of methyl mercury in fish oil or kelp-based products as a result of the conversion of elemental mercury to methyl mercury by many fish and aquatic species by the process of bioaccumulation. At this point of the presentation, I'd like to give you a little background to the United States Pharmacopoeia because I think it's important. USP is an independent non-profit organization which is not part of the US government. However, it works very closely with government agencies, ministries, and regulatory authorities around the world. The mission of the USP is to improve the health of people through public standards to ensure the quality, safety, and purity of medicines, dietary supplements, and foods. USP standards have ensured consistency for drug products throughout the world for close to 200 years and are recognized in more than 130 countries. These standards, known as United States Pharmacopoeia National Formulary, or USP-NF for short, is a book of public standards for drug substances, dosage forms, compounded preparations, excipients, medical devices, and dietary supplements. The FDA designates the USP-NF as the official compendium for drugs sold and marketed in the US, which must conform to these standards to avoid possible charges of adulteration and misbranding. So what is the USP process to change a standard method or a specification? USP continually revises standards through a unique public-private collaborative process which involves scientists and industry, academia and government, as well as other interested parties. The American Chemical Society was one of the interest parties who worked with USP to update Chapter 231. I lead the Heavy Metals Task Force on the ACS Reagent Chemicals Committee. The test for heavy metals was described in USP Chapter 231, which was first introduced in 1908, which is very similar to the ACS test. The test relies on precipitation of the metal sulfide in the sample and compares the color intensity to a lead standard. USP felt that it was time to replace Chapter 231 with methodology that reflected modern test methods and updated toxicology data. So let's briefly explain USP Chapter 231 in greater detail. It is based on sulfide precipitation of the analyte elements using an organosulfur compound and assumes that all heavy metal analytes behave in a similar manner to a lead standard which samples are compared to. It is semi-quantitative at best that was initially intended to detect a small group of heavy metals, but there was no clear definition of which individual elements the method was detecting. It is well recognized to have many limitations, so it's pretty amazing that it has survived over 100 years. So what are these limitations? This slide shows the colored sulfide precipitates of 12 different heavy metals. Imagine if all 12 elements are present in a sample, how difficult would it be to compare the color to the lead standard, which is circled in red. Some of its known limitations include it cannot detect individual heavy metals. Typically 5 ppm is the limit of detection. It's very labor intensive, and the interpretation of the color varies with the experience of the analyst, and it also needs a large sample size of approximately 5 grams to work. So let's take a look at the timeline to change this method. It began in 1995, where a USP Pharmacopoeia Forum report identified issues with the Chapter 231 method. Then in 2000, an article in the Journal of Pharmaceutical and Biomedical Analysis proposed ICP-MS as an instrumental alternative. In 2004, an article in the same journal showed major recovery issues with USP Chapter 231 compared to ICP-MS. However, it took approximately 12 years for various expert committees, expert panels, working groups, and stakeholder meetings to implement these proposed changes. Yeah, USP definitely works extremely slowly. In January 2018, all USP chapters and ICH Q3D guidelines were finally approved and adopted. So how did the USP and the ICH propose the design and scope of the implementation of these new PDE limits? They decided it was important to have a chapter specifically for pharmaceuticals only, which they called Chapter 232. They wanted to separate dietary supplements from pharmaceuticals since they are regulated differently in the US and other regions around the globe. They call this Chapter 2232. They wanted to simplify the harmonization process between USP and ICH by separating Chapter 232 and Chapter 2232 limits from the analytical procedure, which they described in Chapter 233, which focuses on the plasma spectrochemistry technique, ICP-OES or ICP-MS, microwave digestion procedure, and a full list of validation protocols. Let's talk more about the classification of elemental impurities. This is a slide of PDEs defined in Chapter 232 in microgram per day for three different drug delivery systems: oral, parenteral, and inhalation. But this time I would like to emphasize the first column, which is the class of the elemental impurity. Class 1 includes the classic heavy metals, lead, arsenic, cadmium, mercury, which are known human toxicants that have no use in the manufacture of pharmaceuticals. These should be evaluated at all times. Class 2A elements have a high probability of occurrence in the drug product and also should be monitored at all times. Class 2B elements have a reduced probability of occurrence related to the low abundance, and as a result can be excluded unless they're intentionally added during the manufacture of the drug product. Class 3 have relatively low toxicities by the oral route of administration, but could warrant consideration for inhalation and intravenous routes. So what elemental impurities could potentially end up in the drug products? They identified 4 potential sources. Elements intentionally added in the formulation of the drug substance. Examples of these include platinum group metal catalysts, which are often used in the organic synthesis of some drugs. Elements that are not intentionally added and are potentially present in the preparation of the drug product. Example of this might include talcum powder, which is primarily used for tablet compression. Talc is basically magnesium silicate that is mined from the earth, so it can potentially contain high levels of certain heavy metals. Elements that are introduced into the drug substance from the manufacturing equipment itself. Examples of this include the leaching out of chromium, nickel, and iron from stainless steel mixing containers or storage vessels. Elements that have the potential to be leached into the drug product from the plastic storage containers or closure systems. So basically the bottom line was that regulators require the pharmaceutical industry to have a comprehensive understanding of elemental impurities in its drug products throughout the manufacturing process. So with this as background information, let's now take a closer look at the sources of heavy metals in cannabis. So there are 3 major potential sources of elemental contamination derived from the growing of cannabis and the production of cannabinoid products. They are from cultivation sources. First point to emphasize here is that cannabis and hemp, when grown outdoors, are known as hyperaccumulators of contaminants in the soil and have been used as phytoremediators to clean up heavy metals and radionuclides from toxic waste sites. So if there are high levels of elemental contaminants in the soil, they will likely end up in the plant. Other cultivation sources include other types of growing medium, fertilizers, pesticides, waters, nutrients, and growth enhancers from growing cannabis indoors. The second one, process or manufacturing sources, including extraction and purification procedures, cutting, blending equipment surfaces, mixing containers, storage vessels, plus edible product recipe ingredients and tablet or capsule formulations. And the final source are delivery sources, which include devices such as vaping pens, inhalers, patches, and dropper bottles. So let's take a closer look at what elements could potentially be of concern in growing cannabis and the manufacture of cannabinoid products. First of all, it should be emphasized that if cannabis is grown indoors under more controlled conditions, the plant typically has lower levels of heavy metals. However, if it is grown outdoors, there are about 15 heavy metals found in natural ecosystems, and contamination from industrial activities such as mining, metal refineries, petrochemical plants, power plants, and cement works, etc., that could be potential sources of contaminants in the plant. Also, decades of using leaded paint and leaded gasoline has left its imprint on the environment. The list of elements shown here are ones taken from the evidence in the public domain of heavy metals being taken up at various levels by cannabis and hemp plants. Again, I emphasize that cannabis is a hyperaccumulator that has been used to clean up heavy metal contaminated soil. There is also the potential for concentration of the elemental contaminants during the extraction and purification of the cannabinoids. If it's in the plant and the flower, there's a very good chance it will end up to some degree in the extract. In addition, the manufacturing process could add to this list based on metallic equipment used to grind, extract, and purify the cannabinoids, such as nickel, chromium, and iron from stainless steel containers. To emphasize this point, there have been multiple product recalls over the past 6 to 12 months for elevated levels of heavy metals in extracts and oils. Let's take a look at a few examples. There was a case recently where a CBD oil producer from Florida was forced to recall a CBD tincture by the FDA because it had 10 times higher lead levels than the maximum allowable for Florida, 5 microgram per gram instead of 0.5 microgram per gram. 13 strains of medical marijuana sold at a licensed medical center in Michigan were recalled because they were contaminated with high levels of cadmium and arsenic. Medical cannabis regulators in Maryland issued an advisory to notify patients of potential lead contamination of cannabis liquids in vape cartridges following exposure to heat inside the device. There have been many more examples like this. So let me finish by summarizing my thoughts on future regulations for heavy metals in cannabis. The current list of 4 heavy metals, lead, cadmium, arsenic, and mercury, is clearly inadequate. However, the 24 elements required by the pharmaceutical industry would probably be an overkill. However, it's absolutely critical that the cannabis industry has a much better understanding about the many potential sources of heavy metals in its products, particularly from the growing medium and soil conditions. I think that elemental contaminants derived from the cultivation and the manufacturing process would warrant regulating an additional 12 elements. So a future federally regulated list of potential heavy metal contaminants in cannabis could include the big 4, plus the additional 8 elements required by Maryland and New York, plus an additional 4 elements based on evidence in the public domain, making a total of 16. A number of standard groups have listed other elemental contaminants in addition to the classic big 4 heavy metals to monitor in cannabis. This slide shows a summary of 4 of those groups involved in writing methodologies for measuring heavy metals in cannabis. This is clearly telling the industry that at some point they need to be monitoring more than the big 4 to ensure consumer safety. In particular, I'd like to draw your attention to the list of 13 toxic elements in the list cannabis QAP program, which is developing certified reference materials for cannabis and hemp. And finally, more information on this topic can be found in a white paper I wrote, which was recently published in Analytical Cannabis Online, which is referenced in this slide. I thank you for your time, and I welcome your questions at the end of this training session.
Hello, everyone. Welcome to this AOAC training and education session focused on heavy metals and cannabis. My name is Kaveh Kahen. I'm the chairman and CEO of Sigma Analytical Services. And in this presentation, I'm going to talk about heavy metal analysis in cannabis and hemp flower and derived products. And I'm going to provide a Canadian perspective on this subject. But before that, let me talk a little bit about Sigma Analytical Services. Sigma is a full-service cannabis quality control and R&D lab focused on many aspects of cannabis testing under GMP standards. Sigma is headquartered in Toronto, Canada, and it operates both in Toronto and also in Santa Marta, Colombia. In addition to quality control, Sigma offers a number of other services, including contract R&D, consulting services on GMP, and also cannabis lab design and setup, method development, and validation. Let me give you a quick overview of my presentation. I'm going to talk about the chemistry of cannabis and its complexities. Then I will switch to cannabis quality control requirements. And then I'll focus on heavy metal analysis, and we'll discuss some practical considerations about heavy metals. And then at the end, I will talk about heavy metal concerns in Canada's legal cannabis market. The chemistry of cannabis is complex. And as you know, if you look at the cannabis flower samples, There's a large amount of cannabinoids, flavonoids, terpenes, pigments, fats, waxes, and that ends up being about 50% or more of the flower sample. Something as complex as this makes any kind of analytical measurement quite challenging. In addition, cannabis these days comes in many different shapes and flavors. Flower is obviously the most typical sample type that exists, but these days we are actually seeing lots of growth in what we call in Canada Cannabis 2.0, which is the extracts, distillates, edibles, beverages, and other products, even toothpaste. And these different types of matrices present unique analytical challenges when it comes to heavy metal analysis or any other types of analysis. So how to regulate cannabis? That's a big question because flower is obviously regulated in legal markets because it's inhaled and smoked by the users. But when it comes to the derived products and Cannabis 2.0, as I mentioned, sometimes cannabis falls under food and beverage products. Sometimes it's a nutraceutical, and it could even end up as a pharmaceutical and an API. And obviously regulations are quite different when it comes to these different categories. But in general, if you look at the regulatory requirements for cannabis in different jurisdictions, basically there are two types of requirements. The first category is the main ingredients, which is typically cannabinoids and terpenes. And the second category is impurities. When it comes to cannabinoids, obviously CBD, CBDA, THC, and THCA are pretty much required by law, by regulations in every jurisdiction. And in some jurisdictions, we have seen that CBN has been added to the list. Terpenes, on the other hand, are not required in every jurisdiction, but a number of producers in Canada, where it's not required, actually ask for terpene analysis as well. When it comes to impurities, there's a long list. Heavy metals, which is the subject of this talk, is one of them. But pesticides, aflatoxins, residual solvents, microbial contamination are also part of that picture. And in this talk, as I mentioned, we are going to focus mostly on heavy metal analysis. So why so many regulations? And that's a question that sometimes people that are new to this industry ask. The main reason is that prior to legalization, many types of pesticides were used to improve yield and profits that are very harmful to human beings. Cannabis was grown in facilities that had lead-based paint or other contaminants in soil, and frankly, no one cared. And in the legal world, obviously, that's a big concern. And for cannabis extraction or cannabinoid extraction, solvents were used that were harmful, and the residues were not fully removed. And those were basically the main concerns why we have many regulations today around cannabis. When it comes to heavy metal analysis, I want to provide this overview. So the complex chemistry basically gives us a lot of matrix interferences that we need to deal with. And because of that, there is a big need for microwave digestion to make sure that we remove and get rid of most of that matrix. So in fact, in our labs, I think every sample that goes for heavy metal analysis has to go through a microwave digestion stage to remove that matrix. Having said that, different types of samples may require different digestion methods. And in many cases, those methods need to be validated and make sure that the recoveries are good and the accuracy and precision and everything is there. So that's an additional challenge that's introduced by different types of matrices. Heavy metal contamination from packaging materials is a big concern. And we have seen this obviously in vape pens quite frequently, because there's a lot of metallic parts that could end up leaching to the actual cannabis product. There are no standard methods or sample prep methods available, but USB 232, 233 is generally followed. In Canada, pretty much every producer is following USB 233 to 232 for inhalable pharmaceutical products, and those limits are requested and enforced. However, the focus is on Big Four, and arsenic, cadmium, lead, and mercury are the ones that the regulators and the producers are concerned about. However, we have seen high levels of other heavy metals, chromium, nickel, cobalt, and especially in vape pens, and that's the result of, again, leaching from the packaging material and the actual vape pen into the cannabis product. So if we look at the regulations, as I said, different jurisdictions are applying different levels, but in Canada, it's basically the inhalable pharma products levels, which is 200 ppb for cadmium, 500 ppb for lead, 200 ppb for arsenic, and 100 ppb for mercury. And across the board, these are the levels that are applied. Now, how do we prepare the samples? Typically, as I mentioned, it's microwave digestion. A small amount, about 0.5 gram of sample, is digested with nitric acid and hydrogen peroxide. HF is added sometimes, depending on the sample type. And dilution happens after digestion. And gold is added typically to stabilize mercury, as you all know, because mercury tends to stick to sample introduction parts. And this is a typical digestion program that works pretty well for gummy products. As you can see in this picture, you get a pretty clear digestion using this program. But as I mentioned, when you get new types of samples, depending on the matrix, you may require developing and validating a new sample preparation program, digestion program, and making sure that the recoveries are good. The ICP-MS method that we typically use is a very short method, actually, that focuses on the Big Four, as I mentioned, arsenic, cadmium, mercury, and lead. Kinetic energy discrimination mode is chosen. In this mode, helium is used in the collision or reaction cell to reduce interferences, and in this case, it's especially on arsenic, because arsenic at mass 75 is interfered by argon chloride, and argon chloride must be reduced to the levels so that arsenic can be detected accurately and precisely. And for that reason, we use the KED method. This method works pretty well for pretty much all types of cannabis products. So finally, some observations that I can share with you. As I said, heavy metals in cannabis flower samples in Canada typically follow USB 232, 233, and the limits for inhalable products. But in general, we have seen levels that have been always below the requirements, which is good news. To date, we have not observed any arsenic or mercury levels that would require speciation. In other words, total arsenic and mercury they have been below USP levels, so we have not seen the need to do any kind of speciation before ICP-MS. Occasionally, we have detected high levels of heavy metals in hemp samples, and this is typically a problem in outdoor hemp samples from Ontario. We have seen high levels of cadmium, in many cases failing inhalable requirements. But these hemp samples are used typically as raw material for extraction. So again, it's not clear whether inhalable pharmaceutical product requirements apply to these samples or not. The biggest concern by far is high levels of heavy metals that could come from the containers into cannabis products. This is a major issue in some vape pen products because high temperature is used and we have measured high levels of lead and chromium in certain vape products failing USP limits. We have also observed elevated levels of heavy metals in certain cannabis beverages, especially during stability studies. And the source of heavy metal in this case was clearly from the packaging material that was interacting with the actual beverage. High levels of lead and cadmium have also been detected in rolling papers across Canada. We did an internal study and collected pretty much every types of rolling paper that was available. And in some cases, we have seen high levels of cadmium and lead in those products. In addition to heavy metal contamination, many producers routinely ask for elemental analysis of soil to monitor various nutrients, and we are developing those methods for them as well. With that, I would like to thank our collaborator, PerkinElmer, and also Sigma's QA/QC team. And also would like to thank you for your attention and look forward to your questions and discussions. Thank you.
Hello everyone, and welcome to my presentation, Validation of Various Metals in Cannabis and Cannabis-Derived Products by Microwave Digestion and ICP-MS Analysis. I would like to thank Rob and Kaveh for providing a great introduction to the background of metals testing in cannabis. In my presentation, I will look a little closer at the technology, sample preparation, sample analysis, and the validation guidance for the determination of heavy metals in a variety of cannabis and cannabis-derived products. With regards to heavy metals in cannabis products, there is significant press and scrutiny lately on heavy metals contamination in a variety of products. Most notably, recently we have seen many warnings about lead in vape cartridges. As you can see on this slide, there have been a variety of reports of metals contamination in vape cartridges in the United States. So when I talk about cannabis and the related products of cannabis, what do I really mean? Well, there are a variety of forms that can be found on the market, from flour, butter, concentrates, edibles, and ointments. In this slide, I'm showing an interesting compilation of data performed by the Cannabis Control Commission of Massachusetts. This table compares the maximum concentrations of heavy metals in food and cannabis products. Now these are the maximum values found, so they could be anomalies, but do have some indication that there is a significant potential for heavy metals to get into cannabis products. Earlier, Rob did a good job of summarizing some of the action limits for the selected states, so I'm not going to go through all the states and their limits. But as Rob has indicated, most jurisdictions are following some level of ICH Q3D or USP Chapters 232 for guidance. Many states will base their action limits on a 5 or 10 gram per day dosage. Some of the analytical challenges for determining heavy metals in cannabis and cannabis-related materials is the application actually needs a relatively low detection limit. The big four, arsenic, lead, cadmium, mercury, are actually toxic, so they're required at a very low level in the substances. And what complicates the matter even more is that these substances, these products, are quite varied in their makeup, their constituents. Therefore, we use microwave preparation. Microwave preparation gets the samples into solution much more efficiently, and since it's a closed vessel preparation, we don't lose analyte due to volatilization. Another compounding issue is once we've digested the sample and we've got rid of most of the carbon-based matrix, there's still some residual carbon and other inorganic materials left. That will compound the issue when we put it on ICP-MS by causing matrix suppression and interferences. However, with ICP-MS, we use universal cell technology, or the ability to use a gas inside a cell inside the mass spectrometer to reduce or mitigate most interferences. Now, let's review sample preparation. A major component of a good SOP is the sample preparation section. So, detailing how samples are homogenized, weighed, and digested for the case of metals preparation and subsequent analysis. Homogenizing samples, a very important aspect of any analytical procedure. However, for cannabis and cannabis-derived products, due to the variation of the product type, how you're approaching the homogenization is going to be an important aspect of how repeatable your test is. So ensuring you obtain a representative sample, ensuring that it is ground up finely as best as possible so that the reagents during the digestion process are able to have enough surface area to tack your sample and decompose it to the best it can so you have a nice clear solution at the end. Also, the operator should look for volatile compounds in especially mixed distillates and other extracted sample types. And when you look at some edibles or some samples, cryogenic milling process can actually help quite a bit in the homogenization step. For all our analysis, we use the Titan MPS for the digestion of cannabis and cannabis-related materials. The Titan MPS is a top-loading microwave sample preparation system designed to measure both temperature and pressure indirectly so that we don't inadvertently contaminate our samples along the way. Some important considerations when developing your analytical method. The sample matrix, of course. Is it a solid? Is it a liquid? Is it an extract? Are there volatile components? Is it waxy and hydrophobic? All very important considerations. For most states, you need to use half a gram to get a representative sample. They require a half a gram sample. And also for your method, understanding your reagents, understanding really the cleanliness of the reagents and ensuring they're fit for the purpose. So the beauty of microwave preparation technique is that it works on a variety of products. Anything from the actual flour to gummies, oils, butters, waxes, any sort of sample type that you want to homogenize and put in there. Microwave does an exceptional job of putting everything into solution. Again, during your method, you need to consider all these matrix challenges. Very important part of developing your SOPs. So ideally, SOPs would have protocols for handling the variety of sample types that the lab would experience. So a summary of sample preparation is that we weigh some sample, we put it in a digestion vessel and add reagents to it. The digestion vessel is closed and then is put into a microwave. The microwave is ramped up to a certain temperature for a certain time, and then the samples then decompose. They digest inside that closed vessel. And then once that has completed, there is some time to allow the vessels to cool down, and then the analyst will remove the digestate from there and dilute that to volume. An important part of the sample preparation procedure is actually the pre-digestion step. So once we've homogenized the sample, weighed it into our digestion vessel, and then we've added our reagents, There's about a 10 to 45 minute wait prior to capping the digestion vessel and putting it into the microwave. The reason for this is it will allow you to identify very reactive samples, allow initial reactions to proceed when the cap is off, so that allows CO2 to escape, reducing the risk of overpressurization of the digestion vessel. So what we'll see in a cannabis laboratory is this is a variety of matrix challenges. One in particular are various oils and extractions of cannabis, which can be tricky, especially if the person or company submitting that may not let you know what's in there. One example of extreme matrix challenge submitted by a customer to a laboratory is this one here. So this submitted syringe had unknown material in there, just identified as an extraction. What happened was took this extraction, put 0.5 gram into the digestion vessel, and then proceeded to digest it, even with a pre-digestion. What you see is that you get a rupture, and this can happen, especially if using volatile organic compounds that are in the digestate. They can overpressurize the vessel, and you can have a rupture. In this case, the best way to avoid this is weigh that 0.5 gram of sample, volatilize some of the sample prior to digestion. Then it will go into a solution much more easily. So some general rules for very reactive sample handling. Use less sample if possible. Again, most states are requiring 0.5 gram, so that's not really possible. You can allow the full 45 minutes of pre-reaction time, swirl the mixture, promote the evolution of the volatile substances or CO2 that's generated from the reaction. You can add extra water. This extra water will act as a dampening mechanism during the digestion. Or another way is you can add an extra step during the microwave digestion method to extend the time to get to the higher temperature. And last but not least, you can reduce microwave power as well, and again extend the digestion time a little bit to handle these reactive sample types. So in the end, it all ends up the same. So ideally at the end, you have a nice clear solution. There's no floaties or particulate matter suspended into the solution. And ideally, it's close to colorless as possible. There may be a faint yellow, especially with some of the oils. We see a little bit of a faint yellow left in the digestate, which is fairly normal. But the key is that the solution is clear. And on this slide, some more examples of flour being digested, cookies, oils, all being digested. In the end, it should end up clear. All right. On to instrumental analysis now. So why use ICP-MS for this analysis? The reason we use ICP-MS is for the low detection limit required, especially for the toxic heavy metals. ICP-OES in general cannot obtain these detection limits for these PDEs. Graphite AA and potentially hydride AA can hit these DLs, but it's a much more laborious process of preparing the samples for those analyses. So the big 4 heavy metals can be done in about 1 to 2 minutes for ICP-MS, depending on the instrument configuration. Therefore, throughput is also an important consideration when selecting the analytical technique. So as far as high throughput considerations go, we do recommend using a flow injection system, like the high throughput system shown here on the Nexian ICP-MS. It allows for online internal standard addition. It reduces the overall sample to sample time, overall argon usage, because your sample to sample time is reduced, you're getting your runs completed faster, you're getting your customers' data out quicker, and it reduces mercury carryover, which can be a problem in like a continuous flow setup to get mercury to rinse out of the system adequately. So part of the analytical procedure is that we prepare multi-element standards. They need to cover the expected range of the sample types. So typically for the heavy metals, you're in the sub part per billion to double-digit part per billion. An internal standard solution is typically used, and that will correct for the differences in sample matrix from sample to sample and compared to your actual calibration standards. That internal standard is typically added online via the peristaltic pump. So the primary consideration for the analytical method is that the digestion result in heavy matrix. So you need to understand that the acids in the digestates are fairly high, and so we typically want to reduce the loading of those acids and of the whole matrix in general in the ICP-MS. We typically recommend aerosol dilution to reduce that plasma loading. Using aerosol dilution also avoids bench dilutions and a more efficient operation of the ICP-MS. So PerkinElmer's answer to these high matrix samples is our all matrix solution. This simplifies the analysis by reducing the plasma loading due to the heavy matrix. What it does is it introduces a flow of argon into the spray chamber neck and then dilutes the aerosol stream prior to entering the plasma. This results in more efficient ionization, fewer matrix effects, and less deposition on the interface cones. This results in simplified sample preparation and higher quality data overall. Shown here, PerkinElmer's all-matrix solution allows for signal attenuation by using gas dilution. We can actually do over 100 times dilution using AMS, and there's a predictable response across the mass range. For the analytical method on the Nexian ICP-MS, it's pretty simple. It's just one mode of operation. Typically, KD or collision mode is used just by introducing helium into the universal cell of the ICP-MS. This allows for a streamlined analytical method and faster sample turnaround. Okay, now let's get into method validation. So how do you go about validating your method once it's already developed? You're digesting your samples, they're coming out clear, your analytical method is developed on the ICP-MS. What are the next steps? Example here is the Michigan Marijuana Regulatory Agency just published a revised document on February 11th of 2021. Page 14 and 15, it discusses how all testing must meet the standard method performance requirements for the adopted reference methods. In this case, it's referring to United States Pharmacopoeia, USP, Chapter 233, EPA 3052, the microwave digestion procedure, and the determination of heavy metals in a variety of cannabis and cannabis-related products, the AOAC SMPR 2020.001 documents. If you skip to page 33 of this document, it discusses the heavy metals and analytical requirements for cannabis and cannabis-derived products. I'm taking a couple notes here. So Michigan's requiring a half gram of sample. Their limits are derived from USP's Chapter 232 based on 5 gram per day consumption, and the LOQ must be 1/2 the action limit, which makes a lot of sense. So here's the AOAC SMPR 2020.001, the standard method performance requirements for the determination of heavy metals in a variety of cannabis and cannabis-derived products. Here I've spiked out the validation guidance in this document. Essentially, it's recommending you to reference USP Chapter 233. It's saying arsenic, cadmium, lead, and mercury are required analytes. The other metals are optional. I'm not totally a fan of the LOQ because it doesn't really make sense for all analytes. It says the LOQ must be less than or equal to 10 part per billion Hopefully that'll be updated next version. If you look at the Michigan document, it says the LOQ must be half the action limit, which makes a lot more sense to me. And that this method performance requirements are based on concentration range, and that makes a lot of sense. So for lower concentrations, the tolerance for error is going to be a little bit greater than for higher concentrations of elements. So if we look at USP Chapter 233 and look at the validation protocol for that method, it discusses repeatability, ruggedness, accuracy, and calibration information for that method. It discusses that the system should be calibrated at half J and 1.5 J at least. And that indicates half the action limit for that particular elemental impurity, or 1.5 times the action limit for that particular elemental impurity. Repeatability is determined by spiking the material at 1 times the action limit for that element for that particular material. And then ruggedness is the same repeatability study, but performed by a different analyst over a different instrument or a different day. And then accuracy. Accuracy is determined by spiking the material of interest at half the action limit, at 1 times the action limit, and 1.5 times the action limit. And as you see, this will align quite well with the AOAC SMPR method performance requirements for the different concentration ranges of the different elemental impurities in these materials. Here I'm showing you an example J-value calculation. J-value is calculated by dividing the permissible daily exposure limit for a particular element divided by the maximum daily dose and dilution factor for the preparation of that particular material. So for example, mercury has an oral daily dose permissible daily exposure limit of 30 micrograms per day. So if we digested only 0.1 grams of that material and the daily dosage was 10 grams, and we diluted the resulting solution to 50 milliliters, the J value would therefore be 6 parts per billion. What is not defined in USP Chapter 233 is the determination of the LOQ, or the limit of quantitation. So in this example, I'm showing you some data where we did perform the LOQ determination by digesting 10 method blanks, and calculating a standard deviation of those blanks, factor them up by the dilution factor of 100, and then 3.3 times that standard deviation is our limit of detection, and 10 times that standard deviation is our limit of quantitation. So here in this table, I'm comparing our limit of quantitations to the lowest action limits typically found in the various states. These actually come from the Michigan limits that were shown earlier. So as you can see, where LOQs are quite a bit lower than the lowest action limits for these particular elements. Part of every laboratory's quality management system should be regular proficiency testing. We have been participating in the Emerald test of heavy metals since 2018. This year, they now have a heavy metals in oil PT as well as a heavy metals in bud PT. So I would highly recommend PT as a normal part of your method validation and revalidation procedures. In summary, ICP-MS in combination with an optimized digestion protocol is an easy and reliable way of testing cannabis products for heavy metals and other elemental constituents. I wanted to thank everyone for attending my talk today. If you have any questions, feel free to reach out to me at any time via email. Thank you. Bye.
Hi, my name is Ben Armstrong, and I'm the Laboratory Director at Juniper Analytics. I'd like to thank you for tuning in today to the AOAC CASP Training and Education webinar on heavy metals and cannabis. Today, I'm going to be speaking from the perspective of a compliance testing lab that's looking to add heavy metal analysis to its repertoire. Juniper Analytics is an ORLAP and ISO 17025 accredited laboratory that provides compliance testing services to the cannabis industry. We've been in business since 2015 and have been performing compliance testing since the Oregon state regulations for recreational cannabis went into effect in October of 2016. Since that time, we've seen the market expand substantially, with many new products and new regulations being passed for compliance testing. So Juniper Analytics is located in Oregon. We're one of 14 states that has legalized recreational cannabis. However, heavy metal testing is not currently required for state compliance here in Oregon. Colorado, one of the first states to legalize cannabis, added heavy metal testing as a requirement in 2019. Well, the other state, Washington State, is also in the process of currently adding heavy metals as a required compliance test. Many of the other states that have since legalized cannabis do require heavy metal testing, such as California and Nevada, and others that have medical cannabis, such as New York, are also requiring heavy metal analysis. Through this presentation, I'm going to go over some of the steps needed to add heavy metal testing for an analytical lab. First, I'll talk a little bit about the regulations and how they influence the instrumentation that you'll need. We'll talk a little bit about the space and the waste stream requirements for adding this type of inorganic analytical test, and also some considerations for the personnel needed, and finally the timeline for method validation and accreditation. My colleague, Ellen Parkin, just took part in a series of meetings for the Oregon testing work group. And as I mentioned, other states have recently added heavy metal testing as a compliance requirement. Coupled with the reports of heavy metals in cannabis products, as well as the well-documented ability of cannabis to remove heavy metals from soil, it seems very likely that Oregon will soon require heavy metal testing. The current recommendation that the work group is putting forth is to adopt the same action limits used by the state of California for the big four elements, which are arsenic, cadmium, mercury, and lead. And as the industry evolves, and if other elemental contaminants are being identified, there is a potential for this list to expand. That's kind of an important consideration when you're thinking about the instrumentation that your lab may need. So to that point, another important consideration is the variety of sample types that need to be tested and the potential interference that you may get from those samples. To demonstrate this, I wanted to highlight the wide variety of cannabis products that we routinely see for analytical testing in our lab. So these pictures were all taken from samples recently seen here at Juniper Analytics. We test a lot of traditional cannabis flower. That makes up about 60% of the samples that we see. Extracts and concentrates also make up a large part of the recreational and medical markets, which include rosins, live resins, BHO, and other hydrocarbon-based extracts, as well as CO2 extracts and mechanically derived concentrates. There's a huge range of edibles as well, including gummies, hard candies, popcorn, chocolate, and drinks. And more recently, with the continued involvement of the hemp industry, we're seeing a large number of hemp extracts, single cannabinoid isolates such as CBD and CBG, gel caps, creams, lotion, and even pelletized food for animals. So this wide variety of products and the various extraction methods used to create them all pose different and unique challenges in preparing and analyzing these samples for heavy metals. When choosing what instrument you'll need, it's important to consider how many elements you'll need to test for, the detection limits that you need to hit, your sample throughput, and potential interference you may encounter due to the wide variety of sample types that your lab sees. Cost is also a consideration, but I'm mentioning that last as it's essential to have an instrument capable of performing the type of analysis you need to do. Here I'm showing two of the most commonly used instruments for heavy metal analysis. Graphite furnace atomic absorption is good at analyzing a few elements. It normally needs about 2 to 3 minutes per element for each sample, which means that it has a relatively low sample input. The analytical working range is also quite low, but the cost of the instrument is only around $50,000. On the other hand, inductively coupled plasma mass spec instruments can determine more than 73 elements per minute and have a very large analytical working range. This gives for a very high sample throughput, with sample digestion and sample prep probably being the limiting step. The full suite of instruments needed for an ICP-MS cost usually in the $150,000 to $200,000 range. Due to the low detection limits, the potential for sample interference, again due to that wide variety of sample types, and the throughput that many medium to large size labs see, an ICP-MS is usually the preferred instrumentation for performing this type of analysis. It's important to have a dedicated workspace for performing this type of sample preparation. Acid digestion is typically used to ensure that trace metal elements are completely dissolved. For this reason, it's ideal to have a dedicated fume hood to work in, as well as to control the waste stream, as the waste stream from this sample preparation is not compatible with the organic prep methods used for the majority of other analytical analyses, such as cannabinoid or pesticide sample preparation. Due to the variety and complexity of sample types needing digestion, this digestion is usually performed with a microwave sample preparation system. I'm showing here the PerkinElmer Titan MPS microwave sample prep system. And as I mentioned earlier, the sample preparation and digestion is usually the limiting step in throughput for this type of analysis, as the read time on an ICP-MS is so fast. In preparation for adding heavy metal testing, our lab had to take a hard look at the space requirements needed to add this type of instrumentation. Since Juniper Analytics started in 2015, our lab has grown from pretty small to now a medium-sized lab. We have 2 HPLCs for cannabinoid analysis. We're utilizing 2 GCMS systems and 2 LC triple quad systems, along with our sample preparation space, moisture content, and water activity analyzers. And so, along with the fume hood, the digestion microwave, the ICP-MS instrument, as well as the chiller, vacuum pump, auto sampler, and the computer system to run everything, it became pretty obvious to us that we were going to need a new and dedicated space. There's also a strict temperature range and relative humidity needed for optimal instrument performance. So it's a really good idea to make sure that the space for this instrument has the appropriate heating and cooling, as well as ventilation. So when we were looking into adding an ICP-MS, it became apparent that we were going to need this dedicated workspace, which also meant for us, we were going to need a bigger facility. These systems also need to be vented to remove toxic fumes. So it's ideal to find a location that has a room that you can dedicate to housing these instruments specifically from everything else in the lab. Special care had to be taken to ensure that the fume hood, the microwave prep system, the bench, and instrument would all fit without the space getting too hot or cold. And the fume hood is also meant to work for sample preparation and ventilation. But another consideration we had to make was to compensate with makeup air for all the air being removed from that space through the fume hood. So the diagram I'm showing here is a mock-up I made of the design plan for our metals room. And I think that this is about as small as you could make this space. The room we're going to try to use is about 15 feet by 11 feet. So currently, there are no standardized methods for sample preparation for heavy metal analysis. And because there's such a wide variety of products in the market, these different sample types may require distinct digestion methods, and optimization for this process can take some amount of time. Luckily, there's a lot of similar work that's been done in the food and pharmaceutical industries, and we can use these methods as a starting point for cannabis products. Once the system is actually up and running and you're ready to start performing validation and then eventually your production runs, I think the sample prep and analysis will be pretty streamlined. However, because this is a unique prep method with regard to the other sample preparations that we perform in our lab, And I'm kind of assuming that we're going to need a qualified person to handle this analysis along with other functions in the lab. So you might need to think about adding dedicated personnel to just handle this type of heavy metal prep and analysis. After getting the instrument set up, I'm preparing to need at least 4 weeks to perform method validation. And then we'll have to perform 2 rounds of proficiency testing before we can apply to add this method to our scope of accreditation. The Oregon testing work group is kind of offering a recommendation of at least 7 months for labs to go through this process. And again, that will really depend on your region, the type of accreditation body that you have, and the timeline for getting that done. Once you're actually ready to perform production runs, I think that the analysis will be pretty streamlined. But again, because you're going to have this distinct sample preparation technique, you'll have to kind of consider how many matrices you need to optimize your preparation and analysis for. So in summary, it's important to consider the required elements and the action limits in your area when you're considering what instrumentation to use. This type of inorganic analysis requires a dedicated waste stream, a dedicated space and particular consideration for the waste stream that you're going to generate. Some future directions. It may be really important to consider testing for metals in final packaged products, such as once extracts have been added to a vape cartridge. There's a potential to also provide elemental analysis for clients for soil and other nutrients that they're using. And I would just like to encourage everybody to get involved with the AOAC CASP program. I'd like to thank the team at AOAC for organizing this event and giving me the opportunity to present. I'd also like to thank my team here at Juniper Analytics and all of you for tuning into the presentation today. Thank you very much, and feel free to contact me with further questions.